Hello and welcome to the presentation of our group project where we ask the question, how does task difficulty affect recruitment of different brain regions? So our group is called TADA, standing for top-down processing. And so, like I was saying, we wanted to look at how the brain recruited different regions of difficulty of trials. So we were working with the assignments data set. And so the first thing you can see on the top left-hand corner of the screen is a 3D schematic of where the probes were placed in the brain. And then just beside that is a trial schematic showing that the mice had a wheel and they had to turn it to the area with higher contrast. Just beside that, we have a mean firing rate as a time series for different regions based on the NoGo versus the hard trials. So the NoGo trial is when there was no contrast presented to either side of the mouse and they had to remain still in order to get a reward. Using Granger Causality, we looked at the connectivity between different regions of the brain during these different tasks. Basically, we examined functional connectivity recruitment of different brain regions using Granger Causality on these time series data, and we did this in a pairwise manner for all the areas recorded. This produced a causality matrix of p-values, which detailed how likely the activity of one area was to predict the activity of another area for 10 time steps. After this, we used Bonferroni correction on the p-values and corrected for type 1 and type 2 errors using multi-comparisons. And then we only used p-values that were less than 0.01 to construct the functional networks in the bottom right-hand corner. And as you can see, the hard trial seemed to have greater connectivity across regions than the NoGo trial. Then just to the left of that, we compared the number of upstream and downstream partners for each node in this network. And we examined this between these different types of trials, and very interesting features emerged. In general, individual areas have more upstream and downstream partners on the hard tasks compared to the NoGo trials. As such, the hard task has a greater functional connectivity across the brain areas. But does this mean the mice perform better? I'm now going to pass over to Patricia.
To see whether the effect also manifests in the raw data and have a proof of concept with basic statistic measurements, we looked into more detail into the spike trains and the accuracy presented within the Steinmetz dataset. We noticed that when we look at the response accuracy of the different mices, that the hard trials, or the trials we labeled as hard, that the absolute contrast difference is equal to 0.25, were the ones where the mice performed the poorest. The results on all the different tasks were non-conclusive. That means that they varied in such fashion that we couldn't decide whether those effects actually showed a clear pattern and were significantly different. Therefore, we decided that only the hard against all the rest will be grouped for the difficulty level. If you look into the spike trains, you see that the traces of these mean firing rates over the time course of the experiment, only the NoGo and the hard trials were significantly different. We also ask ourselves, how does this differ with different statistical measurements? For example, if you take time compressed measurements like interspike intervals, where you lose the temporal structure of the data, but you have the indication of the interspike intervals, this might tell slightly different stories. In general, what we see here in the middle part, if you compare the coefficients of variations between all areas, or our particularly areas of interest, which were in the primary visual cortex, the primary motor cortex, the prefrontal cortex, the nucleus accumbens, and hippocampal structures, is that you do have a slight increase in the CV, means that the neurons tend to fire more irregularly. But those differences in the coefficient variations are so small that it's not safe to say that this is a clear effect of the task difficulty, or rather just a fragment of the analysis we performed ourselves and the small recording times of 2.5 seconds. As another statistical analysis, we actually looked at the oscillations present in the LFP data also provided in this Steinmetz dataset. And what you can see is that actually you do have phase lockings between the primary visual area and the area called ACA in this graph, which is one of the prefrontal areas we looked at. This means that the time series are not just more predictable of each other, but also in the hard task example are more synchronized between those two brain areas than we initially thought, which is a really interesting correlation we found at the very end of our 2 weeks projects. In general, when we talk about our experience or our insights, we can say that actually getting from spikes to continuous time series data can be really tedious, and we ask ourselves a lot if we apply the tools correctly. On the other hand, Granger causality, as we first initially thought, seemed to be really straightforward to apply. So we wouldn't have a lot of problems, but as we look deeper and deeper into the literature, we realized that there are slightly different pitfalls and special caveats you have to think about when you want to apply a Granger causality. For example, the multiple comparisons corrections, Bonferroni corrections. In the general, when we think back about our project and how we structured ourselves, we have to say that we definitely learned that we should set a limit time for theoretical discussions and project outlines, because we realized if there are multiple people who are working on the same topic and are really enthusiastic of this topic, discussion can be quite long and go on for several hours, because you just tend to go deeper and deeper into the discussions. And at some point you do have to go back to the data. Also, what we realize is if you have such a rich data set as the Steinmetz data set, it can be quite hard to actually do a proper selection of the areas. So we tried to narrow down our data set to areas we found particularly interesting. But then again, what computational areas of the brain are not interesting if you think about how the brain solves the task? So this is also one of the discussion we really had to narrow down and limit in time. To conclude, what we've learned is, or what we've seen also through the NMA, is that we can still do and learn really cool science and perform research during and despite the pandemic. Down on the left bottom, you see our team, which was composed of Junji Hua, Ada Duan, Markus Pleitzer, Daniel, who presented the networks to you, and me. So we would like to thank our pod and the NMA organizers for giving us this opportunity.